This is part 3 of our chapter 8 and 9 PowerPoint. In this part of the chapter, we look at how you measure heritable variation in a population. This falls under the realm of what we call quantitative genetics. And in quantitative genetics, we look at quantitative traits and how evolution works on those quantitative traits and how we expect those kinds of traits to change. So clearly we expect a lot more of our traits to be quantitative as opposed to qualitative. And so this is just looking at how we might conduct a study to look at how we might measure selection and evolution in a population like that. So remember, in the past, we used Hardy-Weinberg to predict allele frequencies and genotype frequencies, and we could ask about evolution and compare frequencies from one generation to the next to see if evolution was occurring. Well, now we're looking at a situation where we don't just have a single allele, we don't just have one type of trait. We might have a trait that has multiple loci, and so we might want to ask questions about how is selection working on height, how is selection working on running speed, and that sort of thing, which our previous model would have not really been able to do for us. So in order for us to understand how evolution and selection are going to be affected, one of the first things we have to do is we have to really sort of tease apart the phenotypic variation. So as you can see from here, when you look at a population, so let's say you go out and you measure lizards in your backyard and you look at tail length and you have let's say a frequency histogram showing all the different lizards and how many of them have tails of different lengths. What you're looking at there is your phenotypic variation, right? You haven't looked at their genetics. All you know is what they look like phenotypically. And what we do with phenotypic variation is we basically break it up into components. And two of the major components we're going to see, you see in this equation here, your phenotypic variation is the sum of your genetic variation. So how much of the variation in phenotype was due to genetics, and then how much of the variation in phenotype was due to environmental effects. And so these two effects together could add up to your phenotypic variation. Now remember, previously when we had talked about variation, we said there's also typically going to be an interaction term. So you could feasibly have another term over here which would look at the interaction between the environment and genetics. We're kind of ignoring that particular term here and just looking at the two major components. Basically, when we talk about heritability, we're asking how much of that is due to genetic variation. So we see a fraction here. So if we take phenotypic variation and put that on the denominator, because that's the whole variation, and then the portion of that is genetic in the numerator, you're going to get some number between 0 and 1. So if there was no genetic component whatsoever, which is hard to imagine, but if there was no genetic component, it would be 0. And then if it was entirely genetic, which again is a little hard to imagine, then it would be 1. So if none of the differences you found between individuals had anything to do with genetics, it would be 0. If it was entirely genetics and no phenotypic or no environmental component, then it would be 1. One of the ways that we can measure this, we can look at this graph that plots the average of the offspring character. So let's say in this case it's height. So for these parents, what is the average height of their offspring? What is the average height of the parents? And then correlate those two things. And so in the correlation, the heritability part of the correlation is looking at the slope of that line. So how much does an increase in the parents' height, in this case, how much of that is indicated by changes in the height of the offspring? So if we have taller parents, we get taller offspring, this line would start to take on a slope. So this first one here, there really is no slope to that line, so that's a heritability of 0. So we do see that there's some variation in both parents and offspring, but those are not correlated to one another. There's no slope. Here what you're seeing is that you're seeing a slope of about 0.5. So we could think of this in terms of about 50% of the differences that we see in height in between offspring is due to differences that we see in mom and dad. And then over here where we have a slope of 1, virtually all of the phenotypic variation that we see in offspring can be attributed to genetic variation that we see in mom and dad. Note, this is one type of heritability called narrow sense heritability. You'll read about some other ones in your book. This is the only one we're going to consider here, is this narrow sense, and it is the slope of this line. So it tells us essentially how much of the variation that we see in a population is due to these genetic differences. So one of the things that they also try to do in quantitative genetics is they want to ask questions about how populations will respond to selection. If you kind of think back about our simulations that we did in chapter 6, we said, if you have really strong selection, our allele is going to go to fixation for like a favorite allele very, very quickly, and then if it's weak, it'll be a little bit slower. Well, we're kind of doing the same thing here, but now we're looking at quantitative traits. So now we're asking, in a population that has this kind of variation, lots of different running speeds or tail lengths like in lizards, how can we measure how that population is expected to respond to selective pressures? So there's going to be a couple of things that it's going to require for response to selection. Sort of like what we saw in chapter 6, one of those factors is going to be the strength of selection. The response that we saw before, remember, a population under very, very strong selection, that allele went to fixation very rapidly, and then if it was weaker, it still went to fixation, but it took it a long time. So that's that response we were looking at there. So here we're going to look at response again, but we're going to look at it for a quantitative trait. Now we're still going to look at the strength of selection as having an effect, but now we have to also think about heritability. So in chapter 6, we really didn't worry about heritability because we just assumed that this was a highly heritable trait and didn't have to think about measuring that degree of heritability. When we get ready to look at this one component, which is the strength of selection, so remember our response to selection is going to be essentially the heritability, and we're going to see that eventually we're going to multiply that heritability by strength of selection. There's actually two different ways we can measure strength of selection. One is looking at what we call the selection differential. What the selection differential is, if you were to take two populations and think of them at before and after selection, and if you were to compare the mean for, let's say, running speed before and after selection, that would be your selection differential. Now, selection gradient is a little bit more abstract than that. What with selection gradient, though, what you're looking at is if we were to look at the change in the relative fitness in those populations. So the next figure will show this a little bit easier, just the definition. This is looking at mice. It's looking at tail length in mice. And so what we see on this part A here, you have the blue population, which is before selection, and then you have this orangish population, which is after selection. You have two different means. So if we were to take the mean of the before population, maybe it's around 9.25, and if we were to take the mean of the after population, maybe it's closer to around 10ish. And so the difference between those two would be that selection differential. What they're looking at over here in part B is the idea of the selection gradient. So now look what we've plotted here. Instead of plotting the histogram that we plotted in part A, we're taking tail length, but we're also plotting relative fitness. Let's look at this second population, and let's just let their relative fitness equal some value. In this case, we're going to let it be 3. Okay, we're going to assume maybe they have 3 times as many offspring as the ones that the other population. The exact values here are not as relevant as looking at the differences in the slope. And so what we have here is, if you look at it, we kind of are saying, let's go to the mean here, which is a little bit around 9ish, and then the mean here, which is closer to 10. And then what we're doing is we're essentially taking the slope of this line. And so the slope of this line is what we call that selection gradient. And so it's looking at, if we go back here, it's looking at the slope of the regression of the relative fitness on phenotype. So here was relative fitness, here's phenotype, and we draw a line between those two sets of data points. That is our selection gradient. So the gradient looks at fitness differences, and the selection differential just looks at the mean difference in the trait itself. Now remember, we said the important thing about measuring strength of selection is that we're going to need it if we want to measure this entity down here, which is R, the response to selection. So how fast or how much will this population respond to selection? Well, it's going to depend. It's going to depend on what is the selection differential between the two, how much difference is there in the mean of the two populations from one generation to the next, and then what is the heritability of that particular trait. We're looking at differences in the two populations and then heritability to get our response to selection. Your book will put together a case study of this information, of this model response to selection using bumblebees. We are not going to go over that particular example. Some of the tools that we have when we want to understand how populations that have these quantitative traits, how they're going to evolve under selection. One of the things is that we can estimate how much of the variation is due to variation in their genes. We can quantify strength of selection by looking at differences in their survival or reproduction. And then again, we can put this together into that response model. This would be this first one would be our heritability, the second one would be strength of selection, and this last one would be the R value, which is our response to selection. And that's going to equal the top two multiplied together. One of the things they mention later in the chapter is to think about how selection might work on multiple traits that are actually also correlated to one another. Not all traits can be acted upon independently and individually by natural selection. For example, let's imagine that selection was favoring tall individuals. So selection's working on you being tall. It's not working on, let's say, your arm length. But your arm length is going to be correlated with how tall you are, we would hope. And so what's going to happen is the arm length trait is going to go along with the body height just because those are correlated traits, right? So traits that are correlated will tend to be under selection together. And in some cases, what will happen is you'll end up with something. What they're trying to show here is if you have selection on many traits at one time, then what might end up happening is that you end up with selection working more on what we might think of as the trajectory of those two. So flashback to physics a little bit, into trig. So what they're looking at here is your beak depth and your beak width are correlated. So typically if you have a longer depth and width, they're correlated. So what they're saying is, let's just imagine that selection is pulling, favoring a certain beak width. So we're up this way on the graph, but it's also favoring this way on the y-axis, and then it's also favoring us to move to the right, let's say on the x-axis. So where this population ends up could be sort of a combination of those two forces. So it'd be very much like when you did trajectories in physics. Where will it end up going? Well, it's going to kind of go in the middle depending on the forces, the strength of those two forces. So if those two forces were equal in strength, we might end up going at a 45-degree angle between them. If one is a little bit stronger, then the angle might be a little bit closer to the one that has the stronger selection. It's just worth considering the fact that it may be that sometimes selection can't work on individual traits alone. It has to work on them in the background in which they exist, and they're going to be correlated with other traits. This is looking at an example of that. If you look at fitness, you're seeing right up here sort of the peak fitness. So this is looking at a multi-dimensional analysis of selection when you're looking at anti-predator defenses. So think about it this way. If you were, let's say you're blending into your background, you are a striped snake. If you are a striped snake and you are going and moving in the direction where your stripes are sort of going with the pattern of the background, you would be favored because you'd be blending in. If you moved across the background and you were striped, you would be sticking out a lot more. So basically what they're saying here is that you had very high fitness in two different conditions. You had high fitness in the striped snakes that went in a straight line. They were striped, but they went in a straight line. They did pretty well. Or if they were unstriped snakes, then they did really well if they changed their direction a lot. Those two things together did really well. If you had the other combinations, if you had a striped snake that did a lot of reversals, or if you had an unstriped snake that went in one direction, they didn't do very well. So you kind of see some different peaks here, and then you see valleys, low fitness values here. So there's a fitness peak here, and there's a fitness peak there, but then we have two low fitnesses in between on these two. And so this is sometimes referred to as a fitness landscape, and these fitness landscapes can really influence the overall outcome of evolution quite a bit. The last thing we're going to look at is modes of selection. You have looked at modes of selection several times probably in ecology and also in Bio 2, so this shouldn't be anything that's brand new to you. There's three basic modes of selection that we're going to look at here, and they all depend on essentially answering the question, who in the population has the highest fitness, and then what's going to be the result of that. Each one of these populations is going to start off, this A curve is exactly the same, whether we have directional, stabilizing, or disruptive selection. Starting histogram is the same. What's going to happen in directional selection is individuals at one extreme of your frequency histogram, so one extreme, either side or one or the other, is going to have the highest fitness. So in the graph that they have here, it's actually, if we look at probability of survival, is increasing with this higher phenotype here. So this one is favoring individuals at this far end of the right hand of the histogram. What's going to happen, part C is actually showing us what would happen in let's say the next generation or several generations later. If we select individuals, if we favor individuals at the far right, really what's going to happen is this graph is just going to move over to the right. We're going to eliminate individuals on the far left because they're not doing well, and we're going to increase the number of individuals on the right because they're doing better. In essence, this population then will move to the right. If you look back here, our mean was around 7, and here it's a little bit, our mean is a little bit greater than 7. Directional selection, the mean of the population has changed towards the phenotype that has the higher fitness. Another thing that has happened here in most cases is that the spread of this graph will actually be less. So this graph is a little bit narrower than our original graph. What does that really mean? It means that we reduced the variation in our population. So strong directional selection will change the mean of the population, but it also can get rid of some of that variation that we have in our population. We tend to see directional selection in populations that are experiencing a lot of change, like new environmental changes for this population. In the middle, we have stabilizing selection. So again, we start at the middle, but now when we ask the question, who has the highest fitness? The highest fitness is going to be those individuals that are near the average. And that's why we see this graph in the middle here for probability of survival. If you're average, you have the highest probability of survival. If you're below or above that average, you have a lower probability of survival. So it's best to be average. It's not good to be on the extremes. Then when we ask a question down here as to what happens in the future to that population, what we're seeing here is it hasn't really changed the mean. The mean is essentially the same thing, but what this one has done is it's really eliminated those extremes even more than what we saw with directional. Stabilizing selection, the mean isn't going to change, but you're going to eliminate those extremes on either side, thus reducing your genetic variation. What's going to happen in stabilizing selection is we're going to see the type of environment that we're going to see stabilizing selection occur in are populations that have been in a particular environment for a pretty long time, so they're already pretty well adapted. That's why the average has the highest fitness. They're pretty well adapted to it, and the environment's not changing. One example of a population under stabilizing selection would be birth weights in mammals. If you think about it, if a female has an offspring that is too small, then her fitness is lower because there's a good chance that that offspring will not survive if it's a low birth weight. The opposite could be true as well. If the birth weight is too high, that there could be negative consequences to the mom for delivering a large baby. We end up having sort of that stabilizing selection that works on those birth weights. The last one we see here is disruptive selection. In disruptive selection, it's kind of the inverse of stabilizing. Individuals that are average have the lowest fitness instead of the highest fitness that we saw over here with stabilizing. These have the lowest fitness, so it's good to be at one extreme or the other. Both extremes have higher fitness. It's the middle individuals that have the lower fitness. So what's going to happen over time is you're going to eliminate that middle part of the graph. You're essentially going to reduce those and you're going to push them to the side. It's too bad they didn't make this graph go out a little bit longer, because what's really going to happen is that this particular graph is going to become bimodal. So if we allow disruptive selection to continue for a long period of time, we're going to have one peak that is going to occur down here at the low end, and then no individuals in the middle, and then we're going to have another peak over here at the high end that we see in disruptive selection. Disruptive selection occurs in environments that we sometimes refer to as patchy. Patchy habitats or patchy environments are ones where you have sort of these discrete entities. So imagine that you have a population of butterflies or moths, and they visit flowers. These flowers that they visit can be either white or red. There's two different habitats basically that they can visit. What happens then is moths that are white can blend in if they're sitting on white flowers, so they have a high fitness, and moths that are red can blend in if they sit on red flowers. If you were to have pink moths, so if we were to have individuals in the middle here and they were pink, those pink moths can't blend in anywhere. They're going to have really low fitness because it doesn't matter where they sit, they're going to get eaten by birds, whereas the white moths can do well on white flowers and the red moths can do well on red flowers. What's going to happen over time is you're going to end up with a population of moths that are red or white, but nothing in between. If disruptive selection is allowed to continue for a long period of time, it might lead to speciation. It doesn't have to, but it can. So imagine now that your moth populations not only sit on the properly colored flowers, but they then also mate at that time. If red are always mating when they're sitting on red flowers, then they're mating with other red individuals. And if whites are doing the same thing, they're mating with other white individuals. Over time, what could happen is you could get that reproductive isolation where red moths and white moths no longer interbred with one another. And then you would be on the road to speciation when that happens. These are some data showing stabilizing selection in a gall-making fly. Basically, what this is showing us is two things that are affecting the gall diameter in a plant. So what is it that determines the, let's just say, optimal size to make a gall? It makes a gall. Small galls, let's put it this way, small galls get attacked by parasites. So these are little tiny wasps that go in there and they tend to select these really small galls. If you're trying to avoid being parasitized by these wasps, then you would make larger galls. However, birds tend to attack the larger galls, what we can see over here. So the parasitism of birds on these larger galls is high. So if it makes them too small, they get hit by parasites. If they make them too large, they get hit by birds, and therefore gall diameter is essentially under stabilizing selection. It's not good to be too small, and it's not good to be too large. What we see is here's before selection, and then here is after selection. So we're definitely getting rid of a lot of that variation. Even though we haven't necessarily changed the mean a whole lot, we've definitely gotten rid of some of the variation that was there. This is an example of data for disruptive selection. Basically, what you see here is that the size of the bird's mandible will dictate partly what size seeds that it can eat. So in this particular case, what you had a population living in an area where there were two discretely different size seeds available to it. And so individuals that could focus on smaller seeds did really well. Individuals that could focus on the larger seeds did well, but individuals that ate those seeds in the smaller range did not do well, and so they perished. And so that's what we see as far as disruptive selection. The last thing that we mention here is we want to think about genetic variation and its role in selection. and then how selection influences genetic variation. First off, let's look at it the one way. What is the relationship between genetic variation and selection? Remember, you have to have genetic variation in order to get natural selection. We are all identical clones of one another. Selection's not working. Variation is very critical to how selection works. So now let's ask from the other perspective. What is it that selection does to genetic variation? In many cases, it gets rid of it, which is really odd because this is the one thing that selection needs to work, and then selection often eliminates it. If you look back at our graphs, whether we had stabilizing selection or directional selection, both of those, if you looked at before and after graphs, there was a reduction in the genetic variation in the population. Populations that are under strong stabilizing or strong directional selection, you'll actually get reduced genetic variation. Now, disruptive selection does tend to increase variation. Remember, because what it does is it kind of pulls that graph apart into sort of a bimodal distribution. It does tend to increase the variation. However, disruptive selection also probably isn't super common because it's very, very restrictive. It has to be that patchy habitat. So stabilizing and directional selection are probably more common than disruptive. If that's the case, if I think that most populations are under strong stabilizing and directional selection out there, then that should tell me that most populations that I would go and study are going to not have a whole lot of genetic variation because they've been losing it through selection. But they don't. They actually have quite a bit of variation. Natural populations, as long as they're large enough, tend to not lose that genetic variation that we would expect them to lose under selection. Then we ask the question, two different things really with selection and variation. One of them is, how do you get new variation in a population? How can we add new variation? And then the other question can be, once you get it, how do you maintain it? This slide is looking at how do we maintain genetic variation. One of the things that we know is that most populations are not going to be in equilibrium. We know that there's going to be mutations occurring all the time, so they're constantly sort of being thrown into the population. Mutation is something that's typically reintroducing variation, not at a really high, super high rate, but it is introducing them. Now, one of the other ways that we might keep variation there is this thing called mutation selection balance. Remember, we looked at that before in terms of, I think we were talking about the cystic fibrosis gene, and we said, why is it that some of these genes like CF and some of the other ones have such a high ratio or a high frequency in the population? How come they have such a high frequency in the population when clearly selection should be working against those traits? And we said that one of the things that can happen is that selection is eliminating the harmful mutation, but then the mutation itself is putting it back in. Selection is pulling it out, but then there's new mutations that keep arising, putting it back in. Then we might have a frequency higher than what we would have expected. Other things can occur, so maybe disruptive selection is one way that we can keep genetic variation in a population. We have patchy habitats and they might be changing. That allows some individuals to do better in different environments, and so we might have polymorphism in a population like the red and white butterflies. The other thing that we also saw that we've looked at before, overdominance. Remember, overdominance meant that your heterozygous had the highest fitness, and if your heterozygous has the highest fitness, you keep both alleles around. One of the things that can keep variation in there is if we have this sort of overdominance type of selection. Another one that we looked at before was frequency-dependent selection. In frequency-dependent selection, individuals that were rare had higher fitness. Think of what happens if you are rare and you have a high fitness, then a trait that is rare increases in frequency because it does better. Like if females prefer to mate with rare males, it does better until at the point where it now becomes common. The other allele has the advantage, and so now it's favored. Remember when we ran our selection experiments before, we said that under frequency-dependent selection, you would essentially get the maintenance of both of those alleles. The other thing that can happen is selection might be occurring in fluctuating environment. So what if what is favored this year is not favored next year? And so this year we favor the red allele, and next year we favor the white allele. And if we do this back and forth quite a lot, what can happen is in the long term we actually keep both alleles. So if selection is not constant, if it's fluctuating, and it can fluctuate over time or space, we might see both of those alleles or multiple alleles being maintained. One of the other things then that we want to think about is how do we generate generate that genetic variation. This is how you maintain it once you have it. What if you start off with a population that is not variable? How do you get that variation? You're going to have a couple of ways. One is, of course, mutation can generate something new in a population. Another mechanism that can generate variation is sexual reproduction. If you have sexual reproduction, you're constantly making new combinations of offspring. It's not just whatever mom looks like or whatever dad looks like. It's these new combinations of traits. Those new combinations of traits can also increase that variation in the population. Sexual reproduction can also, not only can it introduce new variation, but it can help maintain it. So remember, we talked about linkage disequilibrium before and the loss of genetic variation, and sexual reproduction and random mating were two ways that we could overcome that. We'll set those two mechanisms, reproduction, sexual reproduction, and non-random mating, can also introduce and keep variation in a population by bringing about new combinations of traits that the parents did not have. This ends our video on chapters 8 and 9.